Hey, welcome back. Mr. Richardson here. We've got our last notes of Unit 1 to go over today. So today we're looking at 1.4, which are early cells and bacteria, which are a lot like those early cells. They're not a whole lot more advanced than the early cells we would have had on Earth. So early cells on Earth were probably very simple, just a phospholipid bilayer. That's what we covered in class the other day. Those hydrophilic heads interacting with water, the hydrophilic tails going to the middle where it's dry, and that forms a layer. And that's the membrane of all cells we know of today. So those bilayers could have formed around some RNA and some proteins, and that would have been very early cells. That was about 3.8 billion years ago, so quite a while ago. And again, by today's standards, I'm not even sure we'd call them cells. We'd call them alive. They would have been very simple, but it was those very simple cells, again, all based on that phospholipid bilayer we've been talking about, that would have been the first cells on Earth. The bacteria of today isn't much different. I'm going to draw a diagram here of a bacteria, a couple of the important parts. So starting off, first thing we draw is the cell membrane. So all cells are surrounded by this phospholipid bilayer, and that's what the cell membrane is. Again, hey, we know now that is a phospholipid bilayer. That's what that cell membrane is, surrounding the cells. All right, surrounding that, so just outside of that, another circle. Pretty much all bacteria have a cell wall as well. So surrounding their cell membrane, on the outside of that, they have a cell wall. Now this is different than the plant cell wall, made up of different components. And we'll talk more about that later in the year. It's a plant cell walls. Now coming off of the bacteria cell, we'll usually find a lot of these, they're like hair-like structures. In fact, they're so much like hair, their name is actually Latin for hair. So these are called pili. And these pili, these hair-like structures, again, that's the Latin name, that's the Romans called hair, was pili. They're hair-like structures that are used for a lot of different things, including locomotion, grasping. So you got to think wherever you have one bacteria, you're more than likely to have about 1 billion more. And so they're all kind of hanging out, and they use these They use these pili to kind of grasp each other, kind of hook onto each other and kind of hang out together. And they use it for mating. So we often think of bacteria as being asexual, and they are. They tend to just split in half, and now you have two bacteria, but they can mate sexually as well. They can share genetic information with a neighbor, and they usually use these pili to do that. Coming out of the back end here, a more tail-like than hair-like structure. This is called the flagella. I should say these are called the flagella. Flagellum is singular, so these are tail-like structures. And they're used primarily for locomotion. So you can imagine these bacteria, they're swimming around in water. How do they move? Well, they just kind of flap their tails back and forth and kind of push themselves around. And not all bacteria have them. And bacteria don't have them. So it's possible for bacteria to have zero, no flagella. One, sometimes you just see one flagella, or they can have many. I drew three. So my cell has three, but it could be even more than that. Usually, I think you see them here at the end of the back end of the bacteria, but that's not always the case. I've seen them at the back and at the front. I've seen them all around on cells. So cells just completely covered with them, kind of move in all different directions. All right, so let's move to the inside of the cell then. And we'll start with the very recognizable DNA. Oops. There we go, make sure we're on the camera here. So there's the genetic material inside the cell. Our early cells probably had RNA. RNA can do all the things DNA can for the most part, but it's a much simpler and much easier compound. And so more than likely, those earlier first cells had the simpler RNA. The bacteria of today have DNA. We'll discuss kind of the differences there between DNA and RNA a little more next chapter. But something to note about the bacterial DNA is that it's one circular strand of DNA. People, eukaryotic cells, animals, we tend to have several long strands. There's a beginning and there's an end to our DNA. There's a top, there's a bottom. They look like an eye. Bacteria tend to have a circular DNA. Now you might say, hey, that doesn't look very circular. This is circular, but it's kind of the same idea with the rubber band. When you drop a rubber band, it can coil up. So we know that rubber band is circular, so that's what's going on with the DNA as well. It's circular. And bacteria tend to have just one of those circular strands. Next up, I'm going to draw a whole bunch of little dots in here. And these are ribosomes. And their purpose is to make proteins. So we drew a whole bunch of these little dots because, well, bacteria are going to have roughly 10,000 of them per cell. So there's a lot of little ribosomes. I'm not going to draw 10,000 dots, but we can at least kind of load it up there with lots of dots. And kind of important to note, ribosomes are sometimes considered an organelle. And we always think, hey, bacteria, these are prokaryotes, they don't have a nucleus and they don't have organelles. Well, they do though. They have ribosomes, depending on what definition you want to use for organelles. So we should really be a little more exact in saying that they don't have membrane-bound organelles. And again, we'll talk more about that in our next chapter. So bacteria are prokaryotes. In the strictest sense, there are two types of, whoops, two types of cells. There's eukaryotes, which like our cells have a nucleus. And then there are prokaryotes, like bacteria, that lack a nucleus and other membrane-bound organelles, which we'll talk about those later in the year. But in all honesty, prokaryotes, bacteria, these guys, this is their world and we're just living in it. There are a lot more of them than us, and I don't just mean total numbers, but by total species as well. There are two domains of bacteria, prokaryotes. So by contrast, there are three total domains of life. All of life fits into three domains. Eukaryotes, again, that's our types of cells. We're just one domain. And that one domain includes the four kingdoms you might be aware of. So you've got animals, plants, protists, and fungus. That's our four kingdoms of life. If you think about every animal you can think of, every species of animal, and that includes insects as well. I don't know if you're aware, but there are something like 350,000 species of beetle. So that's all animals, all insects, all that. We all fit into that kingdom animal, and we're just one kingdom. And then there's plants. You think about all the many types of plants. And then there's fungus. You think of all the different types of fungus. And then protists is kind of the odds and ends that if you don't fit into one of those other three, fit into protists. Well, we're all the eukaryotes, four kingdoms, tons and tons of life. Prokaryotes have two domains. And I've heard as many as 28 different kingdoms. So that's a lot of kingdoms, a lot of different types of life that are prokaryotic. So starting off, first domain. It's called Archaeobacteria. So archaic, kind of like archaic or ancient. And the reason why we call them that is that they live in extreme environments. Thermodacetophiles, so you know the word philes, loves. Thermo is temperature, so they love heat. And acid is, well, it's acidic conditions. They love heat, they love acidic conditions. Thermodacetophiles, they love to live in places that are 80 degrees Celsius. We think it's hot when it's 80 degrees Fahrenheit. 80 degrees Celsius is like 175 degrees Fahrenheit. So it's incredibly hot and like 2 pH. That's very, very acidic. Your body would start to shut down at a pH of like 6. So going even lower than that is really, really acidic, down to pH 2. But these archaeobacteria love it. Again, we call them archaeobacteria, ancient because they live in extreme environments, probably similar to those on ancient Earth. Really hot, fiery, acidic places, not the beautiful, lovely Earth that we know of today. So that's archaeobacteria. Then the second domain is eubacteria. Now don't be confused by you. I know we have eukaryotes, but this is a bacteria, so this is a prokaryote. And eubacteria they're much more similar to us eukaryotic cells. So they aren't eukaryotes, they're bacteria, but they're much more similar. Run out of room on the paper here. So eubacteria are the ones you're probably more familiar with today. They're the ones that kind of live where we live and really share the world with us. So there we have it. That's the end of Unit 1 notes. We started with water and some of its property and compounds, worked our way into lipids, definitely got the phospholipids, very important. We saw that they form the basis for the cell membrane. There is no cell without that cell membrane. And now we're looking at those very early cells. Hope this helps. We're back with more videos on our next unit. Until then, see ya.